Good morning, students. In the previous class, we discussed about migration of ions and also factors influencing electrophoretic mobility and the classification of electrophoresis like zone electrophoresis and also free electrophoresis. Now, I am going to discuss about the paper electrophoresis, which belongs to the zone electrophoresis. So, here we will use the paper as the supporting medium. So here we will take a thin paper which is used in the chromatography. In paper chromatography we will use Portman filter paper. So that type of filter paper we will use in this process, and we have to spot it with the substances which are going to separate, and we have to influence the electrical charge and going to separate different types of components, and basing upon that we can identify by using staining techniques or the fluorescence techniques. The different types of molecules can be assessed. So first we have to know what is the principle of paper electrophoresis. So here for the paper electrophoresis, so every organism or the substance having different molecular weight. So basing upon the molecular weight, they will be moved from one end to another end. If we use low molecular weight substances, we can use the low voltage for the separation. So because in the low voltage low molecular weight substances will not be moved. So if they have two types of molecules in the taken medium, so low molecular substances can be separated by using low molecular weight. So if we use high molecular weight substances, we can use high voltage, and the large molecules will move faster. So here, basing upon the pH and the other charges, they will be moved from one place to another place. Okay, then next we have to take first filter paper. So the filter paper is made up of 95% of cellulose, and we can use Wortmann filter paper, which is used in the chromatography technique. Okay, so it is slightly absorption capacity. It also have somewhat absorption capacity, and the other apparatus like power pack and also electrophoretic cell. So here the power pack containing the DC current and also controls both the voltage and the current output. So which are having the output of 0 to 500 volts and 0 to 150 ammeters are available in this power pack. So here we have to prepare the electrophoretic cell. So here electrophoretic cell containing electrodes, buffer reservoir, and a supporting media as a paper, and supporting transparent insulating covers for closing the tank. So here we can use platinum for the preparation of the electrodes. The two arrangements of the filter strips are commonly commonly used. These are horizontal or vertical in form, and these both are arranged are equally available, and the choice is usually depends upon our preferences. Okay, then we have to prepare. So here observe the picture. So we have taken the two tanks which is filled with the buffer reservoir. So for the buffer reservoir, we can use different types of buffers basing upon what we are going to separate. So here first we have to fill up with the buffer, and we have to take the paper. Okay, so first we have to take the paper, and we cut into the slender like papers, and for that we in the middle of the paper we can apply the sample by spotting about 0.5 centimeters in the diameter, and going to keep this paper in the sample tank. And special devices are available that we can use capillary tubes for this, and we can be applied before the paper is placed into the buffer. Okay, or after the buffer is taken into the chamber. Then we have to do some procedure. So after the sample application, we have to place these apparatus. So first the buffer is prepared by using different types of pHs and ionic strengths. So for example, for the buffer preparation, if we want to separate the proteins, we can take barbitol. Okay, for the protein separation we can take barbitol buffer or phosphate buffer. For the barbitol buffer we can take or adjust it to 8.6 pH and the ionic strength is 0.05. Okay, we can for if we take a liter of composition of buffer, we can add 10.3 grams of sodium barbitate and 8.4 grams of barbitol. So for this composition we get the barbitol buffer. Okay, so if we want to make phosphate buffer, we can use or adjust the pH up to 7.4 and having the 0.05 ionic strength. And we can for 1 liter composition we have to add 0.6 grams of NaH2PO4 and water, and also 2.2 grams of Na2HPO4. So for the preparation of phosphate. And if we want to separate the nucleoproteins, we can prepare acetate buffer or citrate buffer, and we have to manage the pH up to 4.5, and these both having different ionic strengths like 0.1 and 0.13. So for the acetate buffer composition, for the preparation of 1 liter, we can add 3.5 grams of NaCl and 3.28 grams of sodium citrate, and we have to prepare adjust the pH up to 4.5. For the citrate, we can use 28.646 grams of sodium citrate and 20.6 grams of citric acid. This is the composition for the buffer preparation. And the next one is amino acid preparation. For the separation of amino acid, we can prepare phosphate or Michaelis, both having the pH of 4.6 and the other one is 8.6, and the ionic strength is 0.15 and the Michaelis for 0.16, and we have to use different types of compositions for 1 liter. For the phosphate buffer we can use 20.4 grams of KH2PO4 and Michaelis for 1 liter composition we can add 9.8 grams of sodium barbiturate. So this then solvent is made placed in the tubs or the vertical tubes. Then we can place the paper. So before placing the paper we have to place the glass plates, and after that we can place the paper, and which is just touching the buffer solution, and then we have to add 2 glass plates, or we have to place 2 glass plates on the paper. It should due to the upstand of the paper, and then we have to close it with the close the lid with the perpex plate. Then we have to give the current or the voltage. So frequent observation is necessary. So if it having high temperatures, so it will the paper will be going to charring. Charring means that means the paper will going to be ash. So for this we have to keep this complete equipment in the AC condition. So it will reduces temperature by cooling out the instrument. So after that we have to place these equipment at a temperature of 100 degrees centigrade and wait for 2 hours. So the power and the power switch off power and paper is removed. Once removed, the paper is dried in the hot oven at 100 degrees centigrade temperature. Okay, then now we are going to detect it. So for the detection we will use electrophotogram. That is, one is unknown electrophotogram and the other one is standard one. So unknown electrophotogram means what we are taken from the electrophoresis, that is unknown electrogram, and the another electrogram is standard one that will be already prepared. So compare these two samples, and we have to know what is the bands and what is the substance separated. That is one of the detection assay, that is quantitative assay. And the next one is fluorescence. So fluorescence means the luminescence of the substances by using the stains like ethidium bromide. So we have to add this ethidium bromide to assess the amount of proteins or amino acids. And the next one is fluorescence. It is used for the staining. It is used as the staining, utilized for the detection of different amino acids, proteins, and peptides, and also amino acid derivatives. And in place of fluorescence, we can use dancyl chloride also. The next one is UV absorbance. By using UV absorbance, we can separate proteins and peptides, and as well as we can separate nucleic acids. Okay. The next one is staining technique. So here staining we can use for the separation of proteins. We can use bromophenol blue in the acetic acid, and also we can use dancyl chloride as a fluorescence. Okay. The next one is nucleic acid assessment. We can use methyl green, pyrene, and also ethidium bromide for the separation of nucleic acid. Okay, the next one is polysaccharides. For the separation or identification of polysaccharides, we can use iodine. Okay, then for the separation of lipoproteins, we can use southern black in 60% and ethanol. So, and also for separation of glycoproteins, we can use alkyne blue, and it is very sensitive and visualized techniques. And the next one is detection of enzyme in situ. So enzyme in situ means, so first we have to take a paper, cellulose paper. So it is cutted into, for example, if we take the paper and 20 centimeters length and 10 centimeters width. So two papers are cutted into same measurement. Then we have to take these papers. First, in the first paper we have to going to separate enzymes. That enzymes are present in the one paper. In the other paper we can add some of the enzymes, that means substrates, and we have to going to attach each other. That means taking one paper and attach the opposite paper to each other, that is called as juxtaposed. Okay, so by using this technique we can separate specific type of enzymes, and the paper strip is allowed to in the suitable buffer along with the electrophotogram. The color bands will be appear, which indicates the position of the enzymes. Okay, that is about detection of enzyme in situ. And the next one is quantitative estimation. So the amount, the number of substances, that may be enzymes or proteins, that can be estimated, that is called as quantitative estimation. So number can be estimated. So the color intensity or density of zone may be multiplied with a given area. That's resulting the value would be estimated to concentration of the, to know the concentration of the component. The next one is applications. So these electrophoresis, this paper electrophoresis is used for the estimation of different types of components like proteins, amino acids, and peptides, and also nucleic acids. So these are mainly used for the serum analysis and for the used for the diagnosis of different diseases which are belongs to the serums. Okay, and it is also used for the separation of different proteins like muscle proteins, egg proteins, and also for the milk proteins. And it is also used for the separation of snake venoms, and also this technique is used for the separation of insect venoms also. So the next, it is also used for the separation of polysaccharides, and it is used for the separation of different types of estimations like aminopeptidases and amino derivatives. The next topic is gel electrophoresis. So here we will use gel as the supporting medium and also separate different types of components like amino acids, proteins, DNA, RNA, and also different types of macromolecules. So here we will use different types of gels like agarose gel, polyacrylamide gel, and starch gel. We can separate many of organic substances. So here this is the most important technique compared to the other electrophoresis like paper electrophoresis and pulsar gel electrophoresis. So it is based upon the principle when a potential difference is applied across the electrodes of horizontal electrophoretic tank, it containing gel agarose gel or any other gel, and the biomolecules like nucleic acids are loaded, then that separated according to their molecular size. So basing upon the molecular size they will be moved. This is the basic principle of the paper electrophoresis. So here if larger molecules will move very slowly and smaller molecules will move very fastly. So here we will use different types of electrodes which is made up of the platinums. So here this process may be imagined like a running competition. So that one is thinner, which one is thinner, it will move very fastly, and the other one which is thicker, it will move very slowly. So this is the major or basic principle of gel electrophoresis. So here for the gel electrophoresis, first we have to prepare the gel. How we will have to prepare? So here we will use different types of gels like agarose. So if we use agarose as the gel, we will call it as agarose gel electrophoresis. Okay. So if we use starch as the gel, we will call it as starch gel electrophoresis. Okay. So if we use polyacrylamide as a gel, we will call it as polyacrylamide gel electrophoresis. So first we have to take a glass plate and having the support dock. So here we have to take a casket like tray. Okay. Then and a lot of combs. We have combs also. So first for this we have to take this casket tray and we have to take the tape and completely closed with the tape, which we have two openings. Okay. So that two openings will be closed with the tape, and we have to add the gel, that may be agarose gel or starch gel. So this starch gel or agarose gel is made, first we have to take for example if we take a starch or we can take a agarose. So it is in solid form or semi solid form. So then we have to boil it with water and make it a slurry, and this slurry is taken with a test tube and poured on the slab. Okay. Or poured on the casting tray. Okay. So then made it to stable. Okay. So we have to place the agarose in plain. Okay, without any disturbances. After solidifying, we have to take the comb, and by taking, observe this third picture. So take the comb. It is maybe this is made up, almost these are all made up of the plastic. Okay. So this comb is somewhat inserted into the gel. So in that gel, this comb made some of the wells. Okay. Now we have to take the sample which are going to separate by using the micro injection. So then these samples are added to the column. Then this column, after adding the column, then we have the electrodes. So here anode and cathode, then added to the power supply and on the electrophoresis to move or start moving. Okay. So now the separation will be take places. Then we have two types of electrophoresis. One is vertical and the other one is horizontal. In the vertical we can use starch gel electrophoresis, and the horizontal we can use starch gel electrophoresis. So both these we can use starch or either other agarose also. Okay, so the next one, this is the procedure. Okay, so first set up the gel, and we have to load the buffer and get your sample obtained from the previous purifying technique. Then we have to run the gel. Okay, so after running the gel we have to stain with it. Okay, so um, the the wells, the samples which are present in the wells going to move faster, okay, based upon their size, molecular size. So after stopping the current or voltage, so we have to separate that gel. Okay, so these first we have to open that two plastic jars and the tapes, and we have to separate the gel. So by using stain, and we have to look at, by using UV light, you can observe different types of bands in the below picture. Observe here, we can observe different types of bands which is formed at different distances. So basing upon this, we can identify different types of substances which are already separated in this. So actually in the UV absorption, the luminescent or fluorescence if we use, it will be observed like this, like a luminescent bands. So here, based upon the usage of gel, we are differentiated into three types. So that is agarose gel, starch gel, and polyacrylamide gel electrophoresis. So these are the three different types of electrophoresis. So first one is polyacrylamide gel electrophoresis. So we called it as PAGE. That means polyacrylamide gel electrophoresis. So here it is prepared by polymerizing acrylamide monomers in the presence of methylene bisacrylamide to cross-link the monomers. So here polyacrylamide gel structure held together by using covalent cross bridges. So it is thermostable and transparent, and also strong and relatively chemically inert. So gels are uncharged and prepared in a variety of pore size. So it having different types of pore sizes. Basing upon their pore sizes, they will be separated. So if the gels having, or the molecules having low molecular weight, they will move very faster than the high molecular weight substances. That is, we called it as polyacrylamide gel electrophoresis. So what are the advantages of gel electrophoresis? So here, before going to that, we have to know about column gel electrophoresis and also slab gel electrophoresis. So here, column gel electrophoresis means we can use some of the columns, and also we have to pour the medium which or the gel which are going to separate the molecules what we are going to wish. Okay, so and we are, as we are using the column for the separation of materials, we called it as column gel electrophoresis, and the other one is slab gel electrophoresis. In the slab gel electrophoresis, we can use two different types of gels, that are stacking gel and separating gel. So first we have to take the stacking gel, and upon it we have to take some of the little bit of water sprinkled on the stacking gel, and we have to add the separating gel. So now we have to place some of the wells by using the comb. So then we have to add the we have to add the biomolecules which are going to separate, that may be proteins or amino acids. So then we are injected to or submitted to the electrical current. Then the electrophoresis is going to move. Then when it is going to move, different bands are formed. So basing upon slab, we will use this process that we called it as slab gel electrophoresis. Okay, the next advantages and disadvantages of agarose gel electrophoresis. So the advantages are gel is easily poured and does not denature the samples, and it does not react with the samples which are going to separate, and it is very easy to recover also. This is one of the advantage, and the disadvantages are the gels are melted during the electrophoresis, maybe by high voltage or high current or high temperature, and the buffer can be exhausted, and different forms of genetical materials may run unpredictable forms. So due to unpredictable forms, there is no purified separation of the substances. So this is main disadvantage for the agarose gel electrophoresis or gel electrophoresis. Okay, the next one is applications. So here for the separation of restriction enzymes. So here restriction enzymes means we will use it in the rDNA technology or the other substance, and that means for the largest fragment we will separate it into smallest fragments by using or cutting the substance, that is called as restriction endonucleases or restriction enzymes. So these restriction enzymes digest DNA including genomic DNA or southern blotting transfers. So it often used for the separation of RNA to the northern transfer. So and also for the preservation of DNA and RNA also, the separation of molecule is important. So for that also this electrophoresis is used for the separation, and the analysis of PCR products after polymerase chain reaction to assess for the target DNA which are going to amplify. Okay, the next one is it is used to estimation of DNA quantitatively and qualitatively. Okay, then and that means how many number of DNAs are present and how many number of DNAs are in correct form, that are called quality and quantity of the DNA. Here DNA of the quality of DNA is assessed by observing the absence of streakings or the fragments which do not be contaminated, that is called as quality of DNA. And other techniques are really very important for the separation, including DNA fingerprinting. So here we will use beta mercaptoethanol and sodium dodecyl sulfate for the agarose gel electrophoresis. So here we have many applications like separation of different types of amino acids and proteins. Next, detection, recovery and estimation. So the gels from a columns are removed by using water from the hydrodermic around wells. The column allowing the gels to be extracted under the gentle pressure. The slab gels are removed by introducing metal plates between gel plates and coaxing plates in apart. And before staining the gels, we have to immerse in a 7% acetic acid to guard against diffusion of separated components. To identify unknown components in a mixture, the electrogram may be compared with the electrophotogram, on which that means the electrophotogram is already prepared and the other one is unknown electrophotogram. We have to compare each other. Okay, these components have been electrophorused under identical conditions. In this respect, the electrophoresis is similar to the chromatography. Other methods are fluorescence and UV absorbance, and also the staining. So here we will use, first one is fluorescence. In this we will use ethidium bromide for the separation of RNA and DNA, and for the separation of proteins we can use fluorocyclamine and also use different types of staining techniques for the separation. And for the staining we can use for the UV absorbance, nucleic acids and the proteins absorbs in the range of 260 to 280 nanometers. And for the staining, we can separate different types of components like nucleic acids and different types of proteins and amino acids. In this we can use toluidine blue and methylene blue, and also we can use the acrylamide orange for the separation of nucleic acids which are immersed in the acetic acid. And the next one is for the separation of proteins. So here we can use bromophenol blue in the acetic acid which is immersed in the acetic acid, and also tansyl chloride for the separation of the proteins, and we can also use the fluorescence which is separation for the amino acids. Okay.